All right, HLs, here we go. Cellular respiration. Feel free to ask questions. This basically, we're gonna learn the detailed steps of cellular respiration. It's easy to understand, but there's just too many things to remember. So that's why I'm recording this, so you can just review our lecture as well. All right. So cellular respiration. The big idea is energy is converted to a usable form. So it's energy conversion. If you remember with the SLs, I said it's a controlled reaction. Okay, well, why is this important? Because sugar, when you burn it, there's a lot of energy. It's going to be very exothermic. So we want to metabolize it in steps, like series of steps, so that we can convert the energy from the glucose slowly into ATP. So it's a chemical energy, glucose, into a storage energy, ATP, instead of just burning it and releasing it as heat. It's going to be wasted energy. So energy is stored in the molecule of glucose. We try to split it, cut it down, hydrolyze, oxidize it, and then we convert the energy from glucose into a storage energy in the molecule of ATP. Okay, so it's just basically energy conversion from chemical energy in glucose to storage energy in ATP. All right, so before we proceed with the different steps of cellular respiration, since you both are in chemistry, so um, we have to know specific types of chemical reactions here. So first is oxidation and reduction. We've kind of heard about this in chemistry, right? Oxidation, I don't know, somebody mentioned, is the loss of electrons or loss of hydrogen. Okay? Oxidation is the loss of electrons or loss of hydrogen. You can use this mnemonic oil rig to remember that. Oxidation is loss, reduction is gain. Okay, and then remember we said that you're gaining electrons. So I'm like, what? How do you become negative if you're gaining? Because what you're gaining is negative. Anyway, so those concepts are important in cellular respiration. Okay, loss of hydrogen or gain of hydrogen. All right. Um, in your book, oxidation also meant gain of oxygen. Reduction is loss of oxygen. Okay, that's another definition, but don't worry about this. What the definition you need to know is these two. Okay, loss of hydrogen and electron for oxidation, gain of hydrogen and electron for reduction. Okay, so that's the first um, chemical reaction you need to know. Number two is phosphorylation. Okay, what is phosphorylation? Phosphorylation simply is um, when you are when you're adding a phosphate to a molecule. Remember, this is a polyatomic ion, PO4 3 minus, right? That was something that you kind of had to memorize before. Anyway, so phosphorylation is you're adding a phosphate group to a molecule. All right. So there's two kinds of phosphorylation. There's substrate level phosphorylation and oxidative phosphorylation. So what is substrate level phosphorylation? Substrate level phosphorylation is when let's say you have this molecule and then you directly add a phosphate group. That's substrate level phosphorylation. Like you just directly add a phosphate to a molecule. Okay. What is oxidative phosphorylation? Oxidative phosphorylation is indirect, meaning you will have molecules that will undergo a series of oxidation and reduction reactions, and in the process, attach a phosphate to an ADP to form ATP. Meaning this molecule, series of oxidation reaction to become a product, okay? As a consequence, phosphate will be added to your ADP to form ATP. So it's kind of like indirect. You're not directly putting phosphate to a molecule. you're converting ADP to ATP as a consequence of these reactions. We'll look at examples of that later. Okay, but anyway, again, substrate level is you have a molecule, you directly add a phosphate to it. Oxidative phosphorylation is things will happen, chemical reactions will happen. As a consequence, ADP will be able to pick up a P to form ATP. Okay, and then lastly is decarboxylation. This is very easy. Carboxylation. D means to remove. The decarboxylation is removal of a carbon. Okay. All right. So this is our three. Okay. These are the three chemical reactions that I'm going to keep saying over and over again: oxidation reduction, phosphorylation, and decarboxylation. Questions so far? Okay. Headache already? Not yet. Getting there. Almost there. Okay, before we proceed to the steps, let's look at the structure of the mitochondria first. Because the mitochondria is where the there's three major steps for glycolysis. Two late, actually almost all of cellular respiration happens in the mitochondria except for glycolysis. So glycolysis is the first step, and this happens in the cytoplasm. Okay. After glycolysis, everything else happens here in the mitochondria. So we need to know the structure of the mitochondria and how it explains the different chemical reactions that happens in the mitochondria. So let's look at the structure first. Let's look at the membrane. So there's two membranes, outer and inner membrane. So this is your outer membrane, this one, there. And then your inner membrane, it's kind of unique. It's seen as fold. So this one right here, there. That's the inner membrane. So the inner membrane is folded there. Okay. Miss, why folded? Every time you see any structure inside the body that's folded, it means the purpose is to increase surface area. Okay, so the inner membrane is folded. It means we're trying to increase the surface area. Why, Miss? Because something really important happens here in the inner membrane. That's the electron transport chain. We'll talk about it later on. Okay, but anyway, so there's two membranes, outer and inner membrane. All right. The space between them right here, you see the space here? That's the, nothing fancy, intermembrane space. There, intermembrane space. Outer membrane, intermembrane space, inner membrane. Miss, that's just a space. Is that important? Yes, I'll talk about it later on. That space is also important because it determines how much a concentration gradient you can form. Okay, next is, what do we have here? Cristae. Cristae, that's just an academic term for the folded inner membrane. So the inner membrane is folded, so the folding is called a cristae. It's kind of the same. Cristae, inner membrane is the same. And, well, there are ribosomes there. Not that important for our topic today. Naked loops of DNA, okay, not that important for our topic today. And then the matrix. Have you watched this movie? I haven't watched The Matrix. I tried to watch it. Gave me a headache. I'm like, what? I can't follow the story. Anyway, matrix. Useless comment there, guys. Ignore me. Matrix is simply the fluid here inside. Okay? The fluid. Kind of like the mitochondria cytoplasm. It's the mitochondria's fluid inside. So, Miss, is that important? Well, yes, because when I say fluid, it means there's going to be chemicals there. There are going to be enzymes there, okay, that will also help in glycolysis. All right, questions with the structure of the mitochondria. Okay, very good. This is how it will look like under the microscope. Miss, do I have to know? Well, in our previous topic, you needed to know how the organelles will look like under the electron microscope. So this is just another diagram here for you to look at. You can clearly see the cristae, the infolding of the inner membrane there, etc., etc. Okay, let's skip this. Okay, another concept. So let's go back. First, I talked to you about important chemical reactions. Then I talked about the structure of the mitochondria. And then now I want to talk about electron carriers. We're going to encounter them as I explain the steps of cellular respiration. What are electron carriers? Nothing fancy. They carry electrons. So they can pick it up and give it away, pick it up, give it away. So they will be carrying electrons around. Either they accept the electron at one stage, and at the end of the stage, they give it away. So they're free to go back and carry electrons again, give it away. Okay, so we have two electron carriers for cell respiration. NAD plus or NAD. The book used NAD. It's the same as NAD plus. And there's also FAD. Okay, so NAD and FAD. This molecule, as you can see, looking at this chemical reaction, it's good that you're in chemistry. This kind of makes sense for you visually. Look at what can happen. NAD can pick up two hydrogen protons, not atoms, and two electrons to form NADH plus H plus. Okay. You can just basically memorize this, but I kind of want you to make sense of this. Look. You have two electrons, right? So if I get one electron and put it here, that will make it NAD. It will neutralize, right? So I'm still left with 2H and one electron, right? So if I put one H here, that's going to be H plus, correct? I still have one electron here. I can neutralize that. So that's remove. That's just NADH. What are you left with? You have this extra H there, H plus. Now that's why it's NADH plus H plus. You can review this part of the video. Okay. If you don't want to, I think it's easy to understand during chemistry. If you don't want to understand, then go ahead. Just memorize it's NADH plus H plus. That's the product. So when NAD plus picks up two electrons and two protons, this is formed. Okay? Now, it picks up, I said it picks up, gaining. So that's reduction. So when NAD picks this up, it's reduced. It's reduced into NADH plus H plus. If this NADH plus H plus at the end of cellular respiration, it's going to lose the protons. It's going to lose the protons and electrons that it picked up. The process is, the reverse is oxidation. So when NADH plus H plus is oxidized, it releases protons and electrons. Get it? Maybe we can repeat for the sake of clarity. When NAD plus picks up protons and electrons, it undergoes reduction and forms NADH plus H plus. NADH plus H plus can lose those protons and electrons when it undergoes oxidation. Okay. This you will appreciate in the electron transport chain. That's a very important concept. Okay, we will refer back to that. But anyway, so that's why we call it an electron carrier, this. Because it can pick up electrons and protons. At the same time, it can lose electrons and protons. It's a molecule, it's a carrier. A simplified equation is this. NAD plus When it's reduced, when it gains electron and protons, it forms this. Okay, NADH plus H plus. When it's oxidized, it will go back to this form, NAD plus. Okay. Question so far? Okay, very good. So, not as important, but we have another electron carrier. We don't see this much. We only see this electron carrier in the Krebs cycle. Whereas your NAD plus, we see it from glycolysis to Krebs cycle to electron transport chain. Okay, this one is only the Krebs cycle. It's also an electron carrier, as you can see here. If it gains 2 protons and 2 electrons, then that's the 2 protons plus 2 electrons. You have 2 atoms of hydrogen, so H2. Okay. All right. Now let's begin the actual story. Once upon a time, you eat food in the presence of oxygen. It can undergo cellular respiration to produce carbon dioxide and water. As a consequence, you form large amounts of ATP. Now, what happens here? Respiration. I don't have a highlighter. No. Okay. So what happens in respiration? So for this afternoon, this is probably going to be the hardest, second to the hardest topic. Hold on. Ate James, can you close my door? I need complete silence because it's the second to the hardest topic that they will learn ever in their IB life. All right. So here are the different steps. Glycolysis, link, Krebs, ETC. Actually, ETC and chemoosmosis together in your book, this is oxidative phosphorylation. So we're going to talk about 4 things. All right, here we go. Let's start with the review of a diagram I showed you last time. So what's happening overall in cellular respiration? At the end of the day, after you get a headache, you can go back to this one. You have a 6-carbon glucose molecule. In the presence of oxygen, you split it into 2, okay? And you remove 1 carbon, you remove 1 carbon. That's it. As a consequence, you generate ATPs. You make lots of ATPs. SL level. Let's go HL level. Let's start with glycolysis. Let's watch an animation first. See it happening before I talk about the steps. Okay, can you see my screen? Animation? Yeah?
Yeah.
Very good. Here we go.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
Okay, so let's look at the detailed steps. Okay, my iPad now, back to my diagram here. Glycolysis. So, I'll highlight what you need to know, okay? Not all, Don't worry about all of these steps. So in a nutshell, what we're seeing in glycolysis is we're basically hydrolyzing glucose, or sorry, not hydrolyzing. uh, Sorry, hydrolyzing plus oxidizing. Did you hear in the video? It started with oxidation of glucose. You've heard those words, oxidation. What does that mean? We already know that oxidation is loss of hydrogen and electron. So basically, this will lose hydrogens and electrons, and it will split into two to form our pyruvate here. Okay, so glucose will undergo oxidation and hydrolysis. So there's going to be a series of steps where that original glucose will lose hydrogens and electrons and will split into two to form pyruvate. That's the short story. Now let's look at the whole story. So we start with glucose. Glucose is converted to glucose 6-phosphate at the expense of energy. Miss, what did you talk about? Look at this. ATP is hydrolyzed into ADP. We're using energy. We're using phosphate from ATP. That's why it became ADP. Where did the phosphate go? To glucose. That's why the molecule is called glucose 6-phosphate. You added a phosphate to glucose. That's an example of substrate level phosphorylation. Okay, there. We add phosphate to glucose. So we use 1 ATP. Okay, next. Glucose will undergo a series of chemical transformations using an enzyme, which you don't need to know. This is the enzyme, phosphoglucose isomerase. You memorize that in college. In high school, it's just converted into fructose 6-phosphate. Okay, next. Fructose 6-phosphate will again undergo substrate level phosphorylation. Look at this. ATP is used and converted into ADP. Where did the phosphate go? To the molecule. That's why now the name is fructose 1,6-biphosphate. Okay, look, there's 2 phosphates now in your glucose. Got it? Okay. Now, quick question from your reading. What is the consequence of phosphorylating a molecule? I think this was in your reading.
It lowers activation energy.
It lowers the activation energy. It makes it more reactive. The reason is once there's 2 phosphates there, it can split. There's, it's easier for it to split because there's a lower activation energy. Very good. That's why we're phosphorylating our glucose. All right. So now we have fructose 1,6-biphosphate. Don't worry about the 1,6, children. 1,6 just tells us the location of the phosphate. Okay, don't worry about it. Well, actually, you can understand. 1,6, it's carbon 1 and the 6 there, end to end. Okay, so fructose 1,6-biphosphate. It's at the first and the end carbon. Carbon 1, carbon 6. Okay, now Juan said lower activation energy, therefore it's easier to split it. Now it's going to split into your, the book used triose phosphate, but you can also use interchangeably. You can say G3P or glyceraldehyde 3-phosphate, triose phosphate, PG3, I think for some textbook. It's the same. Basically, this fructose 1,6-biphosphate will split into 2. Okay, it will split into 2 here. We're here. Okay, then it will undergo a series of chemical reaction. From this one to this one, the chemical reaction will be oxidation and reduction. Look closely. NAD plus is reduced to NADPH. NAD plus is reduced to NADH plus. Why did I say NADPH? Forgive me. I'm in photosynthesis. Go back. In photosynthesis is NADPH. Okay. Now, NAD plus is reduced to NADH plus H plus. Miss, where did the hydrogen and the electrons come from? It came from this. Got it? That's why we said glucose is oxidized. earlier in the video, glucose is oxidized. Glucose will lose hydrogen and electrons. It's talking about this point right here. Your G3P will lose hydrogens and electrons. So remember, in chemical reactions here in cellular respiration, if anything is oxidized, something will be reduced. If something is reduced, something is oxidized. So NAD plus is reduced. Where did the electrons and hydrogens come from? G3P, meaning G3P is oxidized. Okay? G3P is oxidized. I think in your textbook it said triose phosphates are oxidized. At the same time, NAD plus is reduced. All right. Miss, is this important? Yes. Keep in mind, this will be used in the final step, oxidative phosphorylation. That will be used later. Okay. So where are we now? Where are we? Okay. Next. There's this little small arrow that says PI. That's not Philippines. That's not pi in math. That just means a phosphate ion. Okay. When G3P is oxidized and NAD plus is reduced, the molecule is again phosphorylated. We're adding another phosphate here. This is another substrate level phosphorylation. Okay, look at our drawing here. This. There. I look from here to here. You only have one phosphate, right? We're adding another phosphate. Okay, so let's go back here. When G3P is oxidized and NAD plus is reduced, the molecule is also phosphorylated again. Okay. Now, so this phosphorylated molecule, the name is 1,3-biphosphoglycerate. This one right here, 1,3-biphosphoglycerate. Okay, so you can see there's phosphates again there. Okay, now this is an important part. This is where we produce ATP in glycolysis. Glycolysis is going to produce 2 ATP, net ATP. So ADP will be converted into ATP. Where did the phosphate come from? From here. Okay, look. This to this. I'm removing this. That phosphate. Where did I put it? To ADP. To form ATP. Okay. So, because there's 2 of them, we're going to get 1 ATP here and 1 ATP here. Okay. And let's just proceed with this diagram. This is nice to look at. You still have 1 phosphate, right? We're going to remove that again. We're going to remove that again. Look, no more phosphate. Where did the phosphate go? You attach it to ADP to form ATP. Okay, let's go back to my diagram here. So you have here 2 phosphates. Okay, I remove 1, I form 1 ATP. This one is la 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 la. Here, sorry. There's another phosphate, right? I can, oh, what happened? Oh, okay. I can further remove this phosphate, put it to P to form ATP again. So your final pyruvate here will not have phosphates anymore. Where did the phosphate go? You form it here. Okay. Now, end product of glycolysis is pyruvate. Sorry. I want to hide this. How can I hide? That's all right. That looks cleaner. Look, okay. Please look at the diagram. Ask me questions. I want to highlight important steps here. Look at the diagram. I'm not going to use my pen anymore. Okay. Do you see where oxidation of G3P is? Okay, can you point it out? Okay. Do you see where the reduction of your NAD plus is? Yes. We're looking at this one, right? Oops. Do you see where we do substrate level phosphorylation? There in fructose and glucose. Another phosphate in fructose 1,6-biphosphate. So we're just adding phosphates. Lastly, do you see where we remove those phosphates and put it to ADP to form ATP? Okay, we know where it is. That's it. Any questions so far with glycolysis?
Yes.
Yes.
The phosphorylation happens 2 times, right? Whenever the ATP becomes ADP.
Yes. Whenever the ATP becomes ADP, those are your substrate level phosphorylation. Correct. Removal of phosphate to your ADP happens 2 times as well. Here at the end, ADP to ATP, ADP to ATP. So if you look at my diagram here, you look at this right side, the right side, we're forming how many ATP? 4, okay. If you look at the left side, we used 2. So in glycolysis, the net production of ATP is 2. We have a net production of 2 ATP. There's some ATP, okay, sure, we can make use of that. That's why SL said, if our pathway proceeds to an anaerobic cell respiration, we still have that 2 ATP from the glycolysis. That's not a lot, right? Okay. All right, I hope you're well. At the end of glycolysis, at the end of oxidation and splitting of glucose, what did we have? NADH plus H plus, 2 ATP, and pyruvate. List it down, my end product of glycolysis. NADH plus H plus, 2 ATP. Oh, actually, I think in the multiple choice, you need to know how many. 2 NADH plus H plus, 2 net ATP, and 2 pyruvate molecules. This is a summary. I borrowed some of the slides here from bioknowledge.com. You can have a look at that. It's too complicated. I tried to simplify it on the things that you need to know. All right, so they provided this summary. I have yet to see a short answer question on their essay. Usually they ask glycolysis for multiple choice. In case they ask an essay question, these are subject-specific terminologies you can use. Where does it happen? In the cytoplasm. The hexose sugar is phosphorylated. In fact, Christine said it's phosphorylated twice, she said. Okay, and then the hexose phosphate is split into 2 G3P or triose phosphates. Okay, and these triose phosphates will undergo oxidation, right? And whatever it lost will be received by NAD to form NADH. This is, you can also write NAD plus 2 NADH plus H plus. Maybe memorize this. You are amazingly smart students. This was super simplified. This is how the book wrote it also. Reduced NAD. You're chemistry students. You can memorize NADH plus H plus. Okay. Okay. Anyway, at the end of the latter half of the glycolysis, there's 4 ATP produced. However, we've used 2 earlier, so there's a net gain of 2 ATP, only 2 ATP. Also, there's 2 pyruvate. This is my listing. 2 pyruvate, check. 2 NADH plus H plus. check, and 2 ATP, check. Okay? All right, now let's proceed to the next step, the link reaction. I just had to breathe in and breathe out. All right, the link reaction. This is short. This is a short and cute reaction. It just basically, as the name implies, it links glycolysis to the Krebs cycle. Okay, where's the matrix again? It's the fluid inside the mitochondria. So if there's oxygen there, pyruvate will move into the matrix, and this will happen. Tip, tap. I like the diagram for bionology. Look, focus on the yellow circles. This will happen. Oh, what's that? Decarboxylation. Pyruvate is a 3-carbon molecule. This coenzyme A, that's just a fancy name of the enzyme. The enzyme will facilitate decarboxylation. Let's look at that again. Look at the orange enzyme. It will, boom, remove the carbon. Okay? So in the link reaction, pyruvate is decarboxylated, decarboxylated and oxidized. Oh, again, we keep talking about these chemical reactions. Oxidize. What does oxidize mean? Oxidize, let's focus on oxidize. Pyruvate will lose hydrogen and electrons. Where will it go? Looks familiar. It will go to NAD+. Okay, so pyruvate will be oxidized and NAD+ is reduced. Maybe you're going to get confused with this. Oxidation, this is oxidation of pyruvate. So pyruvate is oxidized, NAD+ is reduced. Okay? At the same time, pyruvate is decarboxylated, meaning you remove a carbon from it. So this will undergo oxidation. It will undergo decarboxylation. What's the product? The product of oxidation of pyruvate is this. NADH plus H plus. And the product of decarboxylation of pyruvate is a molecule we call acetyl coenzyme A. Sorry, I have an important text message. Maybe later. All right. Questions here for the link reaction. When you're asked to discuss or explain, talk about oxidation and decarboxylation. Product is acetyl coenzyme A, and you have NADH plus H plus.
This is just one of each, right?
This is happening to one pyruvate. So you can think that after the link reaction, you're going to have 2 NADH plus H plus. Let's list it down. I've never seen this in the test, but anyway, that's okay. You can memorize it. NADH plus H plus and 2 acetyl coenzyme A. All right. All right. There's a summary here. Let me look at what's important. So pyruvate proceeds to the matrix, and there's removal of carbon. Oh, by the way, when you remove a carbon here, carbon plus there's oxygen in the matrix, you form carbon dioxide. So this is where you see these carbon dioxide waste products being formed. Okay. So removal of carbon to produce carbon dioxide. There's reduction of NAD to form NADH. There's removal of hydrogen and electrons in pyruvate. It undergoes oxidation. Yeah, therefore we call it oxidative decarboxylation. I know my kids know that. They got it. All right, the product is acetyl coenzyme A. Okay. I had to call, wow, we're so high tech. I had to call our lab assistant, our Miss Heidi from the library. I'm looking for equipments. Anyway, because the grade 12 chemistry students are taking home material so they can do their IA at home. So we're gonna look at the Krebs cycle. All right. Again. Um, I don't think we need animation. This is pretty straightforward. Okay. So this is still in the matrix. Where did this come from?
The link reaction.
The link. Yes. All right. Look, without talking, see what's happening in the Krebs cycle. Next, 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 next. Repeat. Let's do it again. Focus on the yellow circles. Okay, focus on the yellow circles. Mm. 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 Focus on the yellow circles only. Mm. Mm. And then it will repeat. Again, focus on the yellow circles. Now with my voice. 4 plus 2. 4 plus 2. 6. Minus 1. 5. Minus 1. 4. And then it will repeat. Okay? If you look at the circles, we're looking at the number of carbon. So what's happening in the Krebs cycle is, first, a 4-carbon molecule named oxaloacetate. This do I need to memorize? No. A 4-carbon molecule will bind with your 2-carbon acetyl coenzyme A to form a 6-carbon molecule called citric acid. Do you need to memorize the name citric acid? No. Citric acid will undergo decarboxylation. In the presence of oxygen, decarboxylation, carbon plus oxygen will form that carbon dioxide. Okay. At the same time, this 6-carbon molecule will undergo, excuse me, I'm so excited, will undergo oxidation there, oxidative decarboxylation. That sounds familiar. We've learned what oxidative phosphorylation is. It just means it's oxidized and decarboxylated. So this 6-carbon molecule will undergo oxidation. What does that mean? It means NAD plus will be reduced to NADH. Okay, next. This 5-carbon molecule right here, this will undergo decarboxylation again and oxidation to form a 4-carbon molecule, CO2 right there, and NADH plus H plus. Okay, now this 4-carbon molecule will undergo several oxidations again, the rearrangements or several oxidations. So you form your NADH and FADH2. Here to here is series of oxidations. So you are removing hydrogen and electrons again from this 4-carbon molecule. Where did the hydrogen electron go? Here, to your electron carriers. And so you form NADH, FADH2. At the same time, you form ATP. So you're removing hydrogen, electrons, and phosphate to your molecule. Where did the hydrogen electron go? To this, NADH and FADH2. Where did the phosphate go? To ADP. ADP to form ATP. Now, I think I can already hear Christine's brain, like how many NADH, how many FADH2, how many ATP. Okay. You have 2 acetyl coenzyme A. So you imagine this is gonna happen twice. Okay. So if this circles twice, then you have 2 ATP. Where did the 2 ATP come from? Right here. You have 2 FADH2. Where did it come from? Here. Twice, so 2 FADH2. Where would the 6 NADH come from? Okay, slowly. 1, 2, 3. There. Times 2. So you have 2 ATP, you have 6 NADH plus H plus, you have 2 FADH2. Now on your quiz, future test, I'll probably draw a diagram of something, okay, something like, trying to remember my quiz. Something like, forgive me. I remember it was a circle, and then I will ask you, where is FADH formed? Let's say, don't always assume that it starts from the top. It's a circle. It can be anywhere. So you look at where you have that 4 carbon molecule being converted into a 4 carbon molecule. 4 to 4, that's where you have your FADH. That's also where you have your NADH. That's also where you have your ATP. Like, or I can also ask you, around the circle, where do you form the NADH? So look at where it's 6, 4, 5, 6. You know there's an NADH there. 6 to 5, there's an NADH. 5 to 4, there's an NADH. 4 to 4, there's an NADH. Okay? I don't know why IB asked it in that way. So maybe just referring to this one, make sure you can visually remember where do you reduce your NAD, your FAD, where do you form your ATP? Okay? Now, because we're almost done. Yay, we're almost done. For the next step, we want to focus children on this. NADH plus H plus and FADH2. Okay, we're going to focus on what's going to happen to those molecules. I'll skip this. You can review that. Let's look at oxidative phosphorylation. It's made up of the electron transport chain and chemiosmosis. For this one, we really need animation. So oxidative phosphorylation, it's made up of 2 steps, electron transport chain and chemiosmosis. Electron transport chain is basically the movement of electron from carrier to carrier to carrier to carrier. Okay, how is that important? Because as it moves from carrier to carrier to carrier, it gives energy to these proteins to pump protons out, right? Pump protons out. There. Okay. So that will keep happening until what do you form? A concentration gradient. Lorelai, you can go. I dismissed the SL students. Okay. So next is chemiosmosis. Electron transport chain, again, that's just jump, 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 pumping of protons there, electron transport chain. Next is chemiosmosis. This is in your reading, chemiosmosis. Osmosis is diffusion from high concentration to low concentration. Chemi, or chemical, we're looking particularly at protons, okay? So osmosis of protons. So you have here a buildup of your protons. Therefore, through osmosis, it's just going to flow back into the matrix, right? As it flows back into the matrix, what happens is there, ADP is bonded to another P. Let me just draw a P here, there, to form ATP. So as this flows in, it energizes your ATP synthase to keep forming ATP. All right. Okay. What else do I want to? Okay, here. So oxygen is your final electron acceptor. Now this is important. Remember how electrons flow from here to here to here. If nothing will accept the electrons, the electrons will just build up here, and it will stop further oxidations because there's a buildup of electrons here. Electrons need to be removed. Okay, just so like this can keep flowing. It can keep flowing here. If there's buildup of electrons here, there's no need for electrons to keep flowing. So the final electron acceptor is oxygen. Also, oxygen accepts protons. Miss, is this important that oxygen is also accepting protons? Yes. Why? If this keeps accepting protons, what happens is you're going to have a low concentration of protons inside. So what, Miss? Then there's more drive for osmosis, chemosmosis, because it's low here. So oxygen that plays an important role. It removes the hydrogens so that it can help establish a bigger concentration gradient. It also accepts electrons there so that the flow will continue. Okay, so oxygen accepting protons, accepting electrons will form water molecules. So this is where your molecules, water comes from. Okay, last time we said, like it's amazing how cellular respiration produces water. This is where that water is formed. So oxygen is your terminal electron acceptor. That's it. A final piece of question. Hold on. Can you? I highlighted this in my book. I think this is important. Page 384, under chemiosmosis. In chemiosmosis, 384, do you have your book? Have a look, please. Okay. In chemiosmosis, protons diffuse through ATP synthase to generate ATP. Okay. The mechanism used to couple or to integrate or to mix the release of energy by oxidation to ATP production remained a mystery for many years. But now it's known to be chemiosmosis. So how do we produce ATP? Through chemiosmosis. This happens in the inner mitochondrial membrane. It's called chemiosmosis because the chemical substrate, which is proton, moves across the membrane down its concentration gradient. Yes. So the mechanism for production of ATP is chemiosmosis. And why do we call it oxidative phosphorylation? Let's go back to our definitions. How is this oxidative phosphorylation? Okay. What is oxidized?
To lose electrons for hydrogen.
So FADH2 and NADH plus H plus, they are oxidized. Okay. Oxidative. What is phosphorylated? Right here at the end of the story. ADP is phosphorylated. It becomes ATP. That's why we call it oxidative, here, oxidation, phosphorylation here at the end. Remember earlier I told you substrate level phosphorylation is you directly put phosphate. You directly put phosphate in a molecule. This one is indirect. We were able to add phosphate to ADP as a consequence, as a result of oxidations here. So it's called oxidative phosphorylation. So we're able to form ATP because of oxidation here. Oxidation of what, Miss? NADH plus H plus. And oxidative phosphorylation, what is the mechanism? In case that's asked on the test, the mechanism is called chemiosmosis. We're able to form ATP through a process called chemiosmosis, or movement of these protons, chemiosmosis. Questions? This is recorded, you can review.